Good afternoon. First of all, I would like to thank the organizers, in particular Professor João Saibo and Professor Elvira Fortunato, I'm sorry, for the kind invitation to be here today and tell you about the work we have been developing during the last years at ITKB NOVA that is in line with the different sustainable developmental goals. And so I call this presentation Better Crops for a Sustainable Future. And this is because our ultimate goal, or our main goal, is to make sure that in the future we have food security and, of course, under sustainability. And the graph that you can see here in this slide represents the driving force, or the main driving force of our work. All of us know that the population, the world population, is increasing very fast, and this will imply that by the year of 2050, we need to have the double of the crop production. And as you can see here, for different crops, you have the yield trends, and the dashed lines indicate what we need as a yield trend to reach the double of the crop production in 2050. And the solid lines represent the current yields that we have, that are predicted, that are based on the know-how that we have now. So we have a quite big gap between what we have and what we need for the future. And of course, this needs to be fueled, and the science, especially the plant science, have a main role here. So in addition to the increase of the world population, we also know that the crop production is highly affected by environmental factors, such as heat, drought, cold, high salinity, and also pests and diseases. And so our group has two main areas of research. One of them is involved or is associated with the plant responses to the adverse environmental conditions, and the other one is more associated with the production of biomass, and specifically with the improvement of photosynthesis. Regarding the first one, we have focused on the crop plant rice. And you may ask, why rice? Well, there are several reasons. One of them is because rice for Portugal is a quite important crop. We are the largest rice consumers in Europe. We consume almost 18 kilos per capita per year. The second consumer is Spain with less than 10 kilos. So you can see that we are the largest consumers. We produce 60% of our needs, or we only produce 60% of our needs. And among other reasons are the problems with the rice production in Portugal, like the temperature, the high salinity, and also problems with pests and diseases. And worldwide, it's even more important to work with rice, because as you may know, rice is the staple food for more than 2/3 of the world population, and is highly affected by different stresses, being the most important or the most severe that causes the most effects, the water deficit or drought. And as you can see in these maps, on the left one, you can see the world rice production map, and on the right one, you have the map of the risk for drought. And as you can see, there is a huge overlap between the regions where rice is produced and the regions with the highest risk for drought. So drought is indeed a problem for rice. In our lab, we have shown that we can improve the tolerance to drought in rice. As you can see here, so the left picture shows you that when we increase a key regulator in the drought responses, that in this case is called ice, we have a much higher survival of the rice. And in addition, and even more important, we can reduce the grain losses from 50%, as you can see here, from 90% to 50%. We are also interested in having plants that are more resistant to high levels of salinity and that can grow in marginal lands. So we know that the plants, they have different ways to cope with high salinities. And so if we identify the key regulators of those responses, we can use that knowledge to have plants growing in that marginal lands with the high levels of salinity. It's also known that rice production is highly determined by temperature and light conditions, namely the length of the day. As you can see in the map on the left side, rice production is restricted to some regions of the world, and the number of seasons per year, it's also, for instance, here at the red, it's the only region in the world where we have 3 seasons of rice per year. So it's also determined by these conditions. In addition, it is known that the extreme temperatures like cold and heat, they have a very severe effect on the rice production. So again, if we find the right regulators, that what we are doing in our institute, if you find the right regulators, we can modulate the rice to grow in regions where before was not possible. We have a good example here. We have been working with the key regulator of flowering. So answering to this question, can we modulate flowering requirements? Yes, we do. We can modulate these flowering requirements using this key regulator that, as you can see here, the two plants on the right in the different panels, they are mutated for this regulator and they do not flower. Normally, rice flowers 2 months after sowing. Here, well, not here, but they don't have 2 years, but we have plants with 2 years that have never flowered. And we have also identified all the elements of the flowering pathway, and so we can now use this knowledge to modulate the rice flowering, and so again to have rice growing, especially specific varieties growing in regions of the world that before was not possible. We are also interested on the fortification, and especially on nutritional deficiency. That, for instance, the zinc, that is very important not only for the plant growth, that you can see here, it causes abnormal growth, but also in human beings, the zinc, it's very important. So if we find that what we are doing to identify the zinc transporter regulators, we can engineer rice to grow in regions that are deficient in zinc, and also to increase the levels of zinc in edible parts of the plant, and so being a good strategy to decrease the zinc deficiency in human beings as well. As I told you at the beginning of my presentation, we have 2 lines of research in the lab, one more dedicated to the plant responses to environmental cues, the second one that is more associated with photosynthesis and production of biomass. As you know, photosynthesis is the reaction that allows the plants to use light, water, and carbon dioxide to produce sugars. Sugars that are very important for the plant growth, for the plant production, for the plant yield. And yield is known to depend on the different factors. It starts with the solar energy, with the light interception efficiency, with the conversion efficiency, with the harvest index. It is known that the factor where we have more room for improvement is indeed the conversion efficiency in the photosynthesis. It is already known that there are plants that are the so-called C4 plants that have a more efficient photosynthesis. That is the case of maize, for instance. And you can see here in this picture, when you compare the growth of maize and rice, that is a C3 plant, so it has a different metabolism. Maize grows much faster and produces much more biomass. And why is that? Because it has a more efficient photosynthesis, as indicated here. So it has a more efficient carbon dioxide assimilation, is more efficient also on the use of water, nitrogen, and on the radiation. So we have been involved in a European project called 3 to 4 that aims to identify the right regulators to implement the C4 metabolism, like maize, in the plants like rice that has a C3 metabolism. So improve the rice photosynthesis. More recently, we have been awarded another European project to study to improve the photosynthesis, in this case in rice and potato. So the concept of this project is the following. We have here indicated in the reaction, or the different reactions of the photosynthesis, we have indicated by the letters from A to F the different points at photosynthesis where we can improve photosynthesis. The different mechanisms that can be improved, they are indicated here, but the final goal, of course, is to increase the biomass, so to engineer the plants to have an increased biomass. What have been shown so far is that these individual strategies in model plants, they can improve the photosynthesis. What we want to show now is that this can be used in the crops, and in addition, that we can combine these different processes using mathematical models to identify the ones that can be combined and that can have the best effect, and also synthetic biology to engineer rice and potato with the combination of several processes to improve the photosynthesis. So to finish, I would like to tell you how we see our contribution to the different United Nations SDGs. So we believe we have been contributing, we continue to contributing to have more productive and better crops, to have plants growing in the marginal lands, to have plants with a higher water use efficiency, to have plants that are more adapted to the global warming, but also to train researchers from developing countries. That is something that I didn't tell you, but that is something that we'd like to promote, and that we have in the lab always researchers from developing countries, to promote national and international cooperation, and also to promote the multi-stakeholder partnership. My final slide is to acknowledge all the people that have been involved in these projects. Not only the students, the postdocs, and some senior researchers and colleagues, but also the other national and international collaborators that without them was not possible to achieve our goals. And of course, last but not the least, the institutions involved, and also the funding from the different agencies. So thank you very much.